this computer. Learning in progress. So this month we're going to talk about PostSet. Next month we're going to talk about systems, and I think the following month we really should talk about culinary ideas, because you know there's a lot of interesting ideas out there, and I think people want to start dialing more into the culinary aspects of oysters, more than just, you know, pulling out a cookbook and whatnot. And I think we've been dabbling with that at SPAT, and there's a lot of interest at with our blue hats doing culinary. But I think that's something that we could kind of do formally, a cooking session. And that would be fun.
All in for that.
Yeah. There's Kimmy.
I would love to do cooking session. That would be
she-
fun.
Kimmy.
Hi guys.
She's
Get
been
the
doing a lot of
potatoes in.
baking. I don't know if you can, you know, throw oysters in all the muffins and stuff we've been eating in the brownies, but
Did some savory baking.
yeah.
right?
You know what's funny
Stuffing.
is this whole thing that's happening at the Ram's Head with us is a unique opportunity. It's tough because it's off the beaten track. You know, us mainlanders, the thought of going to Shelter Island is like, you know, I might as well go to Newfoundland. But you know, it's been a lot of fun. They have tremendous food there. I mean, they really have a good chef, and there's a lot of interest in seafood and oysters and all these things. So I think, you know, without beating that concept into the ground, we should be doing that ourselves on our mainland, just kind of getting people more into experimenting with cooking. We've been doing that here at our house, so we have some kind of go-to recipes that. But I think that's a good idea. We'll do. I'll cue that up for June. That we'll do a just so. You know, where people are starting to wander in. I'm not sure how many people are going to come tonight, but we did get, and I thank Darcy for sending out all the emails about all the events that I have planned over at the Ram's Head. I'm kind of excited because I've got a new little musical act that has been a lot of fun. And so we're going to be playing actually every Friday for the whole season. So all of May, June, July, August, whatever, every Friday. And so the idea was to add an hour before we play to do an oyster kind of get together, or not just an oyster get together. I think we're going to do a series of chats with, you know, fisheries people, marine. It's going to be a marine hour. So it won't just be about oysters. It's going to be, you know, there's going to be some history. The guy I play with is a tremendous historian. He just wrote an article about Shackleton, and you know, he's quite a nautical historian. And so I think there's a lot of folks that are going to come out, and it's been, you know, they did a fireside chat, their first one over at the Ram's Head, and it was the director of Downton Abbey and all those kind of period films with his dressmaker, and it was fascinating. It was really a lot of fun. So we're going to try to, you know, promote that for us Marine there, and just get the buzz out. So that's going to be happening. Tomorrow I'm doing a shucking lesson. So Happy Oyster is donating. And by the way, you guys are queued up for being the fourth grower for Thursday night, starting the 26th of May. So that's the weekend, the Thursday before Memorial Day weekend, if you're interested. I'll get, so for folks that haven't been over to the Ram's Head for the Thursday oyster flights, that happens all season long. And it's all of our ex-SPAT people that went commercial, and it was their busiest night every week for the whole season last year. So we're promoting that because it helps our oyster growers and the whole concept of oysters. And then I'm going to be playing on Friday and doing the marine thing. And in general, just so folks know, we're having a tremendous season already. It's what is it? It's the 21st of April. I happened to stop by the Marine Center today and popped into our upwellers, and the ambient water temperature when I left was 57 degrees, which is pretty darn good. We've got a nice batch of oysters in ambient seawater. So we didn't need to, after this weekend, we won't be feeding any of our shellfish anymore, our cultured algae. Everything will be on natural water. And that's pretty good. I mean, we're going to have everything up and running and quite large right at the first week of May. And we already have oysters in our floating upweller. This month we're going to talk about PostSet, just a brief chat about that. Next month we're going to talk about the systems. So if I say upwellers, downwellers, floating upwellers, and you're like, huh, what's he talking about? Well, we'll get to that next month. For folks that have been doing this for a while, they kind of know what I'm talking about. But I also want to mention that we've been doing these more informal sessions, kind of a chat sessions on these the Zoom, but the real nuts and bolts information is all on our website from last year's PowerPoint. So I don't rehash the PowerPoints right now. They're available. They're pretty. There's a lot of information on them, and I don't want to redo it. It's all available, but that's a problem if you haven't seen it. And we're going to talk about PostSet. A lot of that information is there. You can go back on it if you want to ground truth me. And again, it's informal. So if you have any questions, just chime right in and ask away. So basically, we did 2 spawns of oysters this year. And for folks that don't know, Frank, who's there, Frank Kelly, he and Charles Peck were instrumental in growing SPAT's algae and maintaining the algae room. And as they were doing that, they also were instrumental in conditioning our adults, our brood stock, for spawning. And between growing really good algae and retrofitting our system to being a more controlled drip system with peristaltic pumps that really gave them a constant feeding regime. And when I say constant feeding regime, oysters they're very good at what they do. I can't say that they're really intelligent about it. So they're really good at filtering. They're excellent filter feeders. So if they're in a tank and you throw in 50 gallons of algae to feed them, figuring, well, they're gonna eat that as they need it, and they'll have plenty of food for the next 24 hours. No, that's not how they work. They'll filter all that water in a couple hours and basically use what they can in those couple hours and spit the rest out at what we call pseudo feces. So they actually won't utilize that algae very effectively in a big batch feeding. If you just throw a lot of algae in and it's really dark, they might it's not the best way to do it. What you'd rather do is give them a continuous low volume amount over 24 hours. And then they utilize it very effectively, like they do in nature. It's interesting because during brown tide, that they haven't really determined what the problem was with brown tide, if it was just a toxin or if it was just too much algae. It really is like clogging a Hoover vacuum cleaner with too much debris. They just can't handle it. So, the art of broodstock conditioning, in my opinion, is to really be able to pulse that feed to them in an orderly manner. And so they utilize the food to create gonad for spawning. And so we did a spawn on Valentine's Day, and it was a tremendous spawn. It was, I think there were 24 oysters that were conditioned for that. And we ended up with 11 males and 11 females and 2 that didn't spawn. And we got a tremendous spawn. We raised the larvae up. And then we did some controlled experiments on how we were going to set them. And this is kind of what we're going to talk about today. What happens is you've got a larval cycle. And so they go from an egg to a veliger and grow along this trajectory towards what we call an eyed petaveliger. And so what happens is the larvae is rounding out, it's getting more of a gut, it's getting more shell, it's growing in size. And this is over the course of, I'm going to say, 14 days for us. Okay, so if you spawned on Valentine's Day, which was the 14th, by the 14th day, around the 28th, they will have reached their, and actually that didn't happen with us. It happened in 10 days, which was really remarkable. We got our larvae to go from an egg to an eyed petaveliger in about 10 days. Now, what that means is that the larvae are swimming around, they're feeding on algae. And what's really fascinating about them feeding on algae, everybody's always talking about how much water an adult oyster filters. It's like, oh, an oyster filters 80 liters a day of blah, 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 whatever it is that, the statistic is. It's some great amount of filtration. But what a lot of people don't maybe realize, unless you're looking under a microscope, is that these tiny little larvae that you can't see, they're microscopic, are eating algae cells quite rapidly. When they're 1 day old, they're eating about 4,000 to 6,000 cells each a day. And that sounds like a lot. It's like, and when you watch them, you see that they're filtering from day one. It's really quite remarkable. By day 10, 11, 12, they're eating now 50,000 cells each a day. Now that, to me, sounds like a really large number. They're just like, for 24 hours a day, just eating, a cell. I'd have to do the math. You guys can do the math, whatever. 50,000 cells divided by, 24 hours, how many cells that is per minute or second or whatever. But they're chowing. And it's really cool to witness that, because they're certainly not filtering water at the rate that an adult does, but they're filtering from day one. They're doing what they do. And so between having good feed and being good, healthy larvae to begin with, because the adults put on a nice high fat content egg, we got these larvae to reach their final stage in 10 days. Now, in nature, it's about probably more like 18 days. So the reasons why we got them up to that criteria, which I'll go through again, what the criteria is. I think I did last last month, but I'll go through the criteria of what we're looking for to get them to set to become SPAT. That's what we're looking at. And what that is the last larval stage metamorphosing into a juvenile. Okay, that's the process that we call setting or spat fall or post-set, these kind of terms for when they metamorphose. And in nature, it's all the same principle, but the reason why it's taking longer is in the hatchery, we're growing these larvae at 28 degrees. It's about 80, our water temperature is about 80 degrees all the time. We're using 80-degree water, and we're feeding them kind of high test microalgae. So those are two variables that we're manipulating that nature kind of does allow, certainly at a certain time of the year. It certainly doesn't allow it in February. As a matter of fact, there are no oyster spat in nature yet. They haven't spawned yet, which is the coolest thing about what we do, and why your oyster seed, when you're going to get them, which is going to be early this year without question, are going to be so large and growing at such a rapid pace. In nature, they don't exist yet. They have the right now, right at this moment, right as I speak, I can even hear them. Oysters are waking up and feeding like gangbusters to get ready for spawning. They're putting on gonad like Frank and Charlie were conditioning in all of January in the hatchery. Right now, shellfish, that's what they're doing in nature. The temperature is coming up, algae concentrations coming up, but still the temperature is cool. Right now it's 57 degrees. The shellfish are programmed to put on gonadal growth. That's what they're programmed to do. At this temperature, they're putting on gonadal growth for the spawning event. And what's the spawning event is going to happen. It hasn't happened. We haven't gotten that. Tomorrow is supposed to be a glorious day. It's supposed to be like in the 70s or 60s, so, high 60s, probably get to the low 70s. In another couple weeks, you're going to get the first really warm day that heats up the water in shallow creeks really rapidly. And that's going to be a powerful cue for shellfish to spawn. Okay. We manipulate all that in the hatchery. We use heaters. We go from 70 degrees to 80. We elevate the temperature using to mimic what's going to happen in nature. And it works quite effectively. And we went through all the spawning thing a couple months. We've got our Zoom lecture recorded, and we've got the PowerPoint lecture recorded, and all those cues and what's going on in nature is happening. But just to kind of reaffirm kind of the principle of why we do what we do early is right now, there is no natural spat fall, but we've got a couple million beautiful, some of our oysters are already, they would retain in your #1 oyster bag right now, which is crazy. We've never actually had them this big so early. Knock on wood that everything stays alive and it's all going to grow beautifully. So by literally the first week of June, these oysters should be, the size of dimes, and you can get your seed. I wouldn't get them too early. For folks that have been doing this for a while, just tend your stocks that you have, and we'll get them out to you. You can bug me. I don't mind being bugged, and we're gonna be in a position to be quite generous this year, I believe. So, you know, but you don't want to get stuff so small that you just, you know, you can't handle it and it all goes through your equipment. So I know everyone's perched and ready. We'll see how it goes. We're setting up a new floating upweller on Monday that's going to handle a lot of our stock. And it could be that you can get your seed, you know, the first week of June. Normally it's the first week of July. I think we'll be easily a month early. So that's a good sign. And so the reason why this early seed is so significant is because the spring algae bloom can be huge. And it's starting to happen now. If you've gone out to the water, if you've gone over to the Marine Center, you can see that the water is taking on a nice color. It's really coloring up. The water is not crystal clear. It's really starting to bloom. The other thing that you would notice, if you came and you tended your oysters that you've been growing, you'll probably see a fingernail of new shell growth on them. And so, you know, they're definitely feeding and they're growing as we speak. It seems like it's cold. You put your hands in. Yep. Question?
Yeah. So I'm new to the SPAT program. I'm just beginning to learn about it.
Awesome.
And I have a house on Peconic Bay, and I'm able to walk on the beach and I see a lot of oysters. Am I able to help promote them or to help grow them?
When you say you see oysters, how are you seeing them?
On rocks.
Really?
Yeah. And I haven't really seen them this much before.
Well, there's a couple of things that I'd love for you to look at. As you're looking at those oysters, just as a kind of point of interest, see if they've got a little black line that runs up the center of them. Okay. There was a time where you couldn't find a wild oyster set around the Peconics because we had a thing we called it, this was in about 92 and 3, a new disease we called it juvenile oyster disease. And now it's called Roseovarus. It's a bacteria that causes the problem. But right around 92, a lot of our oyster juveniles were all dying from this oyster disease. And so you weren't really seeing wild set anywhere during, you know, the early 90s. And then I remember going kayaking along Broadwater Cove and in that area of Cutchogue and looking at the rib mussel set and oysters on the rib mussels. And they had this little black line is our brood that we use that was resistant to this juvenile oyster disease. So it'd be interesting to see that. You know, you could do different things. The truth of the matter is legally, you're not allowed to take undersized shellfish into your possession. So if you see little oysters, legally, you're not allowed to take them. The good news about SPAT is you don't have to take, we'll give you it. We'll give you the SPAT, you know, but as far as nurturing them, you can definitely, what I would say is if they're on rocks and you're seeing a lot of them and you want to kind of collect them into a mini reef and kind of garden them, you know, oversee them. What would you do? Well, you would keep them from sedimenting over. So if a big storm came up on your reef and it was covered by sand, that'd be a problem. They would probably all die because they can't be buried. If you saw a lot of crabs around them, I mean, you could protect them. You could do it as a little mini oasis.
What I'm beginning to see is I saw a whole bunch of them. And what attracted me to seeing them was I saw a lot of white reflections on rock. And so what that means is that a lot of them, I think, had been eaten by something. Maybe it was seagulls or something.
That's unfortunately, the nature of the beast is that things want to eat oysters, and
Including me.
well, including you. You're going to be fine. You're going to be fine. You're going to eat plenty of oysters, and welcome to the group. We're gonna get you in on eating oysters and on growing your own oysters. The thing is, with the SPAT program, you are 100% protecting these critters from what nature can't provide. So you're providing predator control. You're doing these things to try to, and still predators can get in there. I mean, SPAT members know that a crab can get into anything when they're small, and if you're not tending them, the crab grows bigger and starts eating your stuff. So there's a whole bunch of tricks to it, and as far as wild set, however, I'm intrigued about it because there's not a lot of places where you see a lot of wild oyster set.
I was very shocked to see.
The more that you start seeing, the better the notion is that, and I wouldn't say it's because of SPAT members that are bringing it back. I would say more that it's the oyster industry around Peconics that's bringing it back. Because, I mean, these guys are protecting, talk about a brood sanctuary. If somebody's growing 500,000 oysters in containment, when those oysters spawn, that's a huge plume of larvae that goes out. And because they're in close quarters, you could get a good fertilization rate. So, this new industry, and I call it a new industry because, back in the day of real oyster growing in the Peconics in 1900, which was aquaculture then, it was a completely different thing than what's going on now. And, I don't get a sense that there was a lot of wild set even back then, but I wasn't around back then. So maybe there was. I would be interested for folks to be noticing what's called overset on their oysters. So you've got an oyster that you've been growing and you see a new little oyster that's set on it and is growing. And we do see that. That means they're recruiting into, those are oysters. That's wild set, an overset.
I have seen that.
Yeah. And so those are all good signs. As far as promoting it, there are ways of promoting it. And we could talk about that. There's always ways of doing. One way of promoting it, which is kind of funny, and if you've got family that wants to do something crazy, what you do is you go clamming or oystering or whatnot, or just get some big ones from us or whatnot, and spawn them off of your dock and throw the spawn into the water. There's a way of doing that. We're going to be fooling around with that with scallops all throughout. Hopefully, as a matter of fact, I've got some brood stock coming up in from our scallop long lines, and we're going to be spawning scallops and showing folks how they can do this on their dock. And that's a form of promoting, because if you're spawning at your dock and you're fertilizing the eggs in a bucket and you throw them overboard, you're still getting a better percentage than a fertilization rate than if the oysters just spawn naturally. So
I don't have a dock, but I do have, a small beach that gets washed over in high tide.
Yeah, well, we're going to go through and make sure you come by the Marine Center so we show you all of our crazy stuff. So
okay, I'd like to.
So basically, getting back to this notion of, and it's about what we were just talking about, overset or wild set. What's happening is this: the oysters are spawning, whether they're in the hatchery or in the wild. The larvae, everything is dispersed in the water column. Eggs get fertilized by the sperm. They become a first stage larvae, the trochophore, and they're swimming around. They morph into a veliger larvae, and they're swimming around. And after about, again, in nature, 2 weeks plus, they're swimming around. Now, keep in mind, when I say they're swimming around, they are planktonic and almost what the term would be nectonic. Fish are nectonic. They're swimming around. They're not benthic. They're not bottom. Okay, that's important. They're in the water column, swimming around. Okay. A couple things happen, and we know this because in the hatchery, these are the things we're looking at to get ready for metamorphosis. The first thing that happens is the development of an eye spot, and it's a very conspicuous black dot on the larvae. And I should have queued up all the pictures, but you know, we could show you them live, or you could go back on the PowerPoint, which has all the pictures, or you can Google eye spot if you're taking notes. But bottom line is, oh, as a matter of fact, I do have a video on my desktop I could show you. It's a Cornell video that I think Rory filmed of all the larval stages from egg fertilization to petaveliger, and it's only about a minute long. I could show it at the end of this, and you could see the eye spot. Now, what's interesting about the eye spot is I've never really seen in the literature anybody being very specific about what that eye spot is. I think I know what it is. I think it's very hard to do the analysis on it because it's so small and probably very expensive to tease out and analyze. But if you ask me, that black spot is a concentration of melanin. Melanin's a black pigment. This is a black spot. Melanin is precursor to dopamine. And you would see, we're going to talk about this interesting phenomenon called search crawl behavior. Search crawl behavior, well, we'll get back to that in just a second. Conspicuous eye spot. This is where you get the term eyed petaveliger. Eyed, the eye spot, black dot, develops. Second is pedaveliger. All of a sudden, this thing develops a foot. It looks more like a tongue. It looks very much, you know, all larvae, by the way. If I were to show you a video of larvae, the first thing you would say is, oh, those are clams. Because oysters, scallops, mussels, clams, all the larvae look like little clams. That's the shape of them. They're called umbonate, and they look just like a little clam, but they're just a larval stage of a bivalve, which is kind of cheating. I mean, it's cheating for the clam. How did the clam get, you know, to go the distance and always stay that shape, you know, and everything else turned into some weird thing, especially an oyster. I mean, oyster is kind of, let's say, not amorphous. It's just, you know, irregular, let's say. You know, scallops are pretty regular. Clams are pretty regular. Oysters can be all over the place. There is a quintessential oyster shape. If you're good at tending your oysters, you'll get that quintessential perfect oyster, the way that the creator created it, supposed to look like. If you grow them like me, they look more like a Lay's potato chip or a, you know, some weird thing. You know, it's like, what's that? That's an oyster. Are you sure? Yeah, I'm pretty sure that's an oyster. I've had them every shape. I have them in heart shapes. I save those for my wife. Oh, you made that heart-shaped oyster for me. Well, not really. It was just kind of crushed in the bag there for way too long. But this thing develops, and they call it the foot. Now, keep in mind that in a clam, which also has it doesn't have an eyed petaveliger, it just has a petaveliger. It just looks like a clam, and it stays and it keeps the foot. Clam keeps its foot. An oyster, a scallop, a mussel, they all lose the foot. So but as an eyed petavillager, it's got the little eye spot, it's got the little foot, and it's crawling. If you look under high magnification, that foot is ciliated at the tip. It's a sensory organ, and it's sticking out, and it's sensing the environment. And here's the thing with oysters, and kind of with scallops too, but definitely with oysters. The major difference between an oyster and a clam when it comes to setting is, and a scallop, some scallops do this, is what I'm about to say with that oysters do. When an oyster metamorphoses, it lays down a blob of cement, and it cements to structure. There are some scallops that do that. There are some scallops that actually cement and attach using that same technique. Most of them don't. Most of them have a Bissell thread, like the mussel has a Bissell thread, and it sticks to substrate using this. If anyone wants to make $1 billion, okay, figure out how to make Bissell thread, because Bissell thread is like a glue that sets underwater instantly. There was a student I met in high school, maybe going on to college or whatever. She was studying the Bissell thread of mussels to look at it in terms of sutures. Bissell thread is a remarkable structure, and there's not enough known about it, and there's no really good uses of it, and there should be, because I've watched scallops swim into the water, flap along the side of a tank or whatnot, and then bloop, stick to it. They set out a little piece of Bissell thread and stick, just like Spider-Man. It's like, that's remarkable stuff, that Bissell thread. But with an oyster, they literally, the day they metamorphose, they cement to structure, whatever the structure is. It's also interesting that you're seeing oysters on rocks. Rocks are structure. Are they preferred habitat? Probably not, but their structure. I mean, if they were preferred habitat, you would think you'd see oysters everywhere on rocks, everywhere, you know. What's preferred habitat? Well, it used to be other oysters. I mean, the oyster reefs that were in Manhattan were vast. The oyster reefs in a lot of places, in the Chesapeake. Right now, still, if you go down to South Carolina and spots like there, you get Wellfleet, Cape Cod, has tremendous set on their oyster flats. I mean, their oyster, you talk about oyster set, that's a problem for them. Every oyster they have has 20 more oysters set on it. They're constantly trying to get rid of them because, you know, they're not like our perfect single oyster that has nothing on it. They've got, you know, they look worse than Willie Nelson, for God's sake. They got, or, you know, they got things coming all over them, all over everything. And why is that? I love this thing I saw in the literature that said oysters are gregarious. Well, you know, okay, they all hang out in oyster bars. Yeah, they're all palling up at the oyster bar. But there's a reason for that. And there was a study done on looking at what could enhance settlement of larvae. And so one of them was looking at what's called oyster bath water. Oysters open up and they filter. What's really interesting about it, when you eat an oyster, and a lot of people say, oh, the best part of the oyster is the oyster liqueur, you know? In scientific terms, that's called the extrapellial fluid, okay? And what is it? Well, it's seawater, but it comes in contact with the whole body of the oyster. So when you buy an oyster, which you should never do, you should just, when you harvest one of your oysters and you're eating it, and you open it up, it's been in the refrigerator overnight, and you open it up and there's all this glorious liqueur. Well, you know, before you harvested it, it was just seawater coming in contact with the body, but it comes in contact with the oyster body, and it's, let's say, enhanced seawater. Well, oysters are doing that all the time. They're opening up, they're taking in water, they're feeding, they're filtering, they're passing water through, and they're expelling water. But that seawater, in effect, is slightly enhanced with the body fluids of oyster. And so what this study did was it took adult oysters, put them in a tank of water, let them respire for a bunch of hours, and then set larvae in that oyster bath water to see if it enhanced settlement, which they claimed it did. Now, let's say the claim is good. What that's saying is that oysters, while they're respiring in nature, are constantly sending off a little bit of a, let's say, a pheromone or a cue, saying, well, we're here, that was a good place to be, and we're still here, and it's a good place to be, so come on and set. And it creates this oyster reef. That would be a remarkable thing to happen, but it kind of makes sense in an evolutionary scheme of things. I would say that right now there's so few oysters that you really never get that effect. There are really no oyster reefs to speak of, so it's hard to get that effect. Where there are, like Wellfleet, they get a huge amount of set. Same with South Carolina. You know, there's places that get huge amounts of wild set. And so, you know, would we like that? I suppose we would like that. But I don't think we've ever had that in the Peconics. I don't think it was ever necessarily throughout the Peconics preferred oyster habitat. Some areas, clearly. Horton's Lighthouse used to have a big reef. Oyster Ponds was clearly given the name Oyster Ponds for a reason. It couldn't have been that there were no oysters there and they just said, well, wouldn't it be nice to just call it Oyster Ponds? Maybe oysters will come in. So, you know, it could happen. The fact that folks are seeing wild set is a very good sign to me. And the more oysters in the environment, theoretically, the more wild set you will see. So there's that component of it. Another component is, and here's one that I'm very fascinated. I actually worked for years for my master's thesis on this one concept. And here it is. So search crawl behavior is when this eyed pedovillager kind of stops swimming, goes down to the bottom, and starts crawling around with its ciliated foot, tasting I call it tasting or sampling or sensing the environment. And what's it looking for? Well, here's what I found experimental. You know how in the marine environment, you're working on your boat, you drop your screwdriver off the dock, and 2 days later, you get your kid to go retrieve it. You know, it's like, hey, could you go get that screwdriver down there? It's still, you know, it's still April. So you send him down and he gets his screwdriver, and you got your screwdriver. It's been in the water for like 2 or 3 days. And it's already really slimy. You know, it's not unslimy. It's slimy. It's only been down there 3 days. How come it's so slimy? Well, to me, that slime is gold. We call it biofilm. And we've been doing a lot of experiments with how we set oysters. And what we do is we precondition our substrate that we're going to use. And we're going to talk about this substrate in a minute. Is that a pickaxe underneath your cupboard? It looks like a pick. No, it's an airplane. Okay. Don't mind me. I had a pickaxing bamboo all afternoon. So it looked like a pickaxe to me. It's like, what? You got bamboo on your property? It's a little airplane. Okay. Don't mind me. I'm regressing. But that biofilm, he's going to get the airplane. He's going to play with the airplane while we're recording. Look at that. That's very cool. You got your airplane model there. There it is. Beauty. It's not a pickaxe. It's an airplane. Very nice. Okay. So this biofilm has been analyzed because other researchers were looking at biofilm, and this whole concept of neurotransmitters. Okay. So I got my master's thesis looking at L-DOPA and epinephrine for enhanced settlement of larvae based on some research. It turns out that search crawl behavior is cued on by dopamines. And now some researchers analyzed biofilm to find a bacteria in the biofilm that was what we call a dopamimetic. It was precursor of dopamine. Now we're getting back to that eye spot being melanin. All of these dopaminergic compounds, in my opinion, play a significant role in larval settlement. Now, I spent a lot of time doing the actual chemical, making these different doses of pure L-DOPA and it definitely induced search crawl behavior. I've just gotten away from the chemistry part and am more interested in the all natural approach to it. The chemicals, I'll tell you why, because you don't want to really mess with L-DOPA. L-DOPA is the one that opens up certain doors of the brain that maybe we're supposed to stay shut. Maybe you get a peek, but you might not want to leave that door open too long because certain elements might come in. Ask Ken Kesey, or I don't. I suppose there's a lot of folks that have messed around with these things that I don't know. Anyway, I'm doing more of the all natural approach to what nature was after with these biochemicals. But I am somewhat convinced that that is the role they're playing. So in other words, for instance, with scallops, everyone's always saying, oh, eelgrass, all the eelgrass is gone. We'll never get scallops back. And that was, without eelgrass, you can't get scallops back. Well, that's clearly not true, because if that were true, I would never be able to culture a scallop, because we wouldn't use eelgrass in the nursery or in the hatchery to get scallops. But I will say that eelgrass was the perfect medium in the environment for promoting good, fresh biofilm that scallops larvae would set all over. And the reason why is because eelgrass is a true grass, okay? It is our only marine plant in New York. There's only one plant, and it's eelgrass. Everything else is a protist, but eelgrass is a true plant. It's got roots, it pollinates. It's like a land plant, but it's underwater. And it's a grass. And what happens in the winter is it all gets mowed down and it grows fresh in the spring. So as it grows fresh in the spring, it provides new tremendous surface area for, first of all, for fresh biofilm. And then the scallops set on it. And then if you ever noticed older stands of eelgrass, they get pretty furry with a lot of epiphytes and a lot of stuff on them. And, you know, they get a little, and then they're going to cleave off in storms like a lawn. It gets mowed down and it grows fresh. So it's really good medium for scallop settlement. And scallops utilized that. It was surface area. It had a good chemical base to it that they liked, kept them off the bottom. Perfect. Would other media work? Absolutely. It's not quite the same because some of these protists could be longer lived and get a little bit more maybe sedimented or not as fresh. But definitely, it's surface area. And this biofilm, here's what we've been doing. So again, the search crawl behavior, when they're looking along, what they want to sense is a good environment. One thing that they could sense that wouldn't be a good environment would be dead zones, sulfur dioxide, hydrogen sulfide. If you've gone clamming or you've gone along the bay, sometimes you see these patches of white and almost blue-black anoxic zones. I've certainly seen them. During brown tide, they were all over the place because things were dying and rotting on the bottom and causing anoxia because of anaerobic bacteria that use sulfur instead of oxygen. And you get this, sulfury stuff. Sulfur in the environment is a toxin, okay? The one that can survive all of that doesn't seem to care are clams, because you'll see clams in the nastiest black ooze, reeking sulfur smelling. Why? Because they have a periscope to rise. They can rise above it. And they literally do. If an oyster sinks into a noxic black ooze, it's dead. I mean, sometimes you'll find them. I remember going into Broadwater Cove and clamming and with bare feet, and I'd get this cut on the bottom of my foot, and I'd reach down and pull up an oyster the size of my foot. And the reason why that oyster was still there is the bill was just above the sediment, and it would open up. It was acting like a periscope, but if it went underneath, it was going to die. An oyster and a scallop have no way of feeding if they're not epifaunal, above the sediments. They're not infaunal like a clam. Clam goes up and down like Otis Elevator Man, but it's got a siphon, a periscope, sticks it up to feed. And if you're a gooey duck, you can have a 1 foot long siphon. You can really be down deep. Some steamer clams. The razor clam, which can go very deep. So these things utilize the sediments to their advantage by having a siphon. But an oyster can't do that. I'll never get over when SPAT people that are new, not that they're green and don't know anything, but sometimes they're green and don't know anything. And so they say, well, I want to go plant my oysters. Should I bury them? Well, no, you should definitely not bury them, because if you bury them, they're going to die. They can't be under the sediment. And it doesn't, and I'm not trying to knock anybody that doesn't know that. It's just the reason why oysters are so particular about where they're going to set is they're going to cement. And that's it. It's pretty terminal. If they cement in the wrong area, they're either going to be if there's cement in the wrong area, they might not make it. Okay. If they cement in the right area, then they will live and prosper and go on to recruit into the environment. So that's a very, very important choice that they make that day that they undergo metamorphosis, because that's going to be it. If they cement to the talon of a seagull because it happened to be, you know, wading in the water, well, then it's going to be an aerial oyster for a while. If it you know, were to be on the bottom of a boat that wasn't bottom painted, then it's going to live on the bottom of the boat. If it were in anoxic ooze, it's probably going to sink and die. So that's the kind of the sensing that it might figure out. If it finds a rock on your beach and it's got fresh biofilm, it can set there. It can set there. It can set on a bulkhead. I've seen huge oysters set on Port of Egypt bulkheads, and that was creosoted. I saw oysters at Founders Landing on the bulkheads. That was creosoted. How could it set on creosoted bulkhead? Oh, the biofilm. That's old bulkhead, so the biofilm on there can be fine for it, and it sets on it. Might not stay very well, but you know, sometimes it does. Now, what we do is we try in the hatchery. If you ever wonder how you get these oysters that we get, see, we're trying to maximize our yield. So we use what's called microculture. And I've been experimenting. I've got a high school student. Well, he's homeschooled, but he's a 10th grader and or junior or something. AJ has been with us for a couple months now. And he's doing an experiment looking at different culture types. I'm very interested in culture. We have been buying our oyster shell from Virginia only because it comes in I think I bought a 20-pound bag of it, and it gets delivered and it's 500 micron oyster shell, which is what I want. I could make my own. I probably wouldn't. I'd probably make SPAT volunteers do it, and they would all leave. They would be so mad at me. It's like, you know, after a week and a half of pounding and grinding and sifting and sieving and three of your wife's old blenders later, and you get a little cup of this stuff. Believe me, because I've done that. I've done that. And actually AJ, my student, that's what he did. He wanted to try whelk shell. And I said to him, well, you know, you can't get whelk microculture anywhere. You're going to have to make it. So he and his dad walked up and down Cedar Beach, collected a whole bunch of whelks, crushed them with a sledgehammer at the Marine Center. I sent him home over the weekend, and he used his mother's old blender and came back with a cup full of whelk shell for his experiment. And what our experiments are is, and well, as an example, his experiment, it was a nice little experiment. Whelk shell off of the beach, oyster shell from what I got that had been soaked in water for 2 days, salt water, and fresh out of the box. No preconditioning. And we set on the three different cultures. And we have yet to do the analysis. It's for his sake, because that's what's going to get him into college, is doing research like that when he's a junior. By the way, last year's intern, everyone remember Maggie? Last year's intern? Well, she's deciding, she won first prize in the national award for her thing that she did, calibrating the spectrophotometer for algal cell density at the Marine Center. So she won third place at the all-state for the thing. And now she's deciding whether to go to MIT or Cornell, which she got into both. So yeah, it's great for students to be doing this stuff. You know, we didn't get them into there, but it didn't hurt. So he's doing that. But what we found is that this preconditioned, so taking this oyster shell, and it comes, you know, dry in a bag. If you were to set it in water for a couple days and get that biofilm on there, the larvae set a lot better on it. And so you know, these are things that you want to know and you want to share with other hatcheries. I think they must already know that. The other thing is we recycle our culch, and every year that culch maintains a bit of essence, and then when you condition it, it can get better and better and better. And what we're doing is we're taking a 500, this is the coolest thing. And if you ever want to pan for gold, come on by the Marine Center any Monday, Wednesday, Friday for the next month, and you can pan for gold. It's my most fun job. And I don't get to do it anymore because everyone else wants to do it. Everyone else wants to be a prospector. I've been a prospector for 30 years now with oysters, and it's a lot of fun because here's what happens. You've got this larvae that want to set on something. So instead of making them wonder what to set on, you provide them with kind of a premium habitat. So what we have is 500 micron oyster reef. It's shell chips ground to 500 micron. And the reason why I do that will come up in a second. We soak it in the water for a couple of days, get a little bit of that biofilm on there. Now we set them in a tank. We set the water at 80 degrees. We feed them premium algae, and we dump 1 million larvae into this container. And we hope for the best. And I tell you something, it's really miraculous to take, I said we did a spawn on Valentine's Day, and we had 22 oysters that spawned, 11 of them that were female. So 11 egg-bearing adults to give you like 2 or 3 million seed. It is pretty good return. I wish my investments these days were doing as well, which they're not. But I'm told not to look at it or talk about it. It'll come back. Okay. Well, so I should be investing in oysters, but I'm investing in SPAT members. So we did really well. And what panning for gold means is this. They set on 500 micron shell chip. Metamorphosis, what happens with metamorphosis is that the oyster's crawling along, it says, I want to set here. And then what happens is another remarkable phenomenon. What happens with the oyster larvae is that epinephrine dumps into their system internally, and it causes irreversible morphological changes. It instigates metamorphosis. They go from a larval stage to a juvenile stage, and the change there is explosive. So the other thing I didn't mention about this, you had an eye, you had a foot, you also had a size of about 250 microns. That's the size of this larvae. So it's about 1/4 millimeter when it goes to set. And when it sets the next day, it doubles in size. It puts on explosive new shell growth. And I've got pictures and I've got things, or you can come and look and we'll do this and I show you, but it's really phenomenal. I think last month I sent a little video of a 1-day-old PostSet oyster, and you could actually see the heart beating. I've got that queued up. All of this is on our website. But the point being that, well, I'll get back to that in a second. The point being that in just a few days, this thing is already 500 microns. So all they have to do is grow to 700 microns, and theoretically, they're bigger than the culch that was 500 microns. So you can pan for pure oysters by taking all these different sieves and just sieving the oysters to size, and it leaves all the old culch behind until you've got pure oysters. And it's actually a lot of fun, and we have some panning to do. And we also constantly sizing our oysters. So we're constantly sieving, and to me, it's a lot of fun because we're going to, on Monday, start counting how many we have. I don't like to count really tiny PostSet because you need reading glasses, and I refuse to be caught wearing glasses in front of anybody. Oh, here they are. We got some somewhere. But when they're big enough to count, you take a volume of them and you can kind of know how many oysters you have. I really don't know how many oysters we have right now in our possession because I haven't done any counts yet. So Monday is going to be sieving and counting sessions. If anyone wants to get in on that, you have to be very careful. I get neurotic when people drop even a single oyster. So I'll show you how to do it. So you never drop an oyster, and you've got beautiful oysters. And it's just very interesting how this larvae finds its habitat, sets on it, and you would never see it in your oysters. That's what makes a single oyster, because if you look down if you set them on a little bit larger of a shell trip so you could see it, it never goes away. It's always at the hinge. This little tiny dot was where it cemented the day that it set, and it's always there. Sometimes it sets on a mussel, and it's set on a mussel and it grows on the mussel. Sometimes it sets on another oyster. Sometimes the larvae set on top of another larvae. That's where you get a double oyster, which you will get occasionally. It's just an oyster setting on another oyster that day. And so that's all fine. What we also did this year, which we did last year as well, but we're going to do this every year, is we did what's called a remote set. So we took a bunch of different shell, oyster shell, we used some scallop shell. We put them in a tank, and we threw the larvae in the tank, and now we have spat on shell. And if you were here at the beginning when Frank and I were talking, he witnessed this whole thing, and it's really remarkable how well our oyster larvae set on this different shell. And what we're going to do with the shell, we did this last year, and a lot of the interns got involved and SPAT members got involved. Now you have a shell with oysters set all over it, and you walk along the banks and you kind of plug them in to the sediments, and they'll grow a little bush of oysters. So that's something that you can do actually to promote oysters growing on your beaches and things, is to start putting spat on shell on different places. And that's what Building Reefs and the Billion Oyster Project and a lot of these projects that Greg Rivara is doing with he's got a spat on shell, pretty large machine. Well, we're doing the same thing, but we're just doing it on a smaller scale, and it's kind of fun. It's not the same thing if I were to say to you, well, there's 100 oysters that set on this oyster shell. So here, you get 10 shells. There's your 1,000 oysters. You probably wouldn't like that so much. You rather have a 1,000 perfect single oyster seed. It's a different thing that we're doing. Spat on microculture is how you would get single oysters for aquaculture for like industry. Some people didn't. The West Coast never did that. They always did spat on shell, grow them on the flats, throw them all over your flats, and grow clusters of oysters. And when you go to harvest them, you harvest them with that that pickaxe underneath your desk, or literally a masonry hammer to get an oyster, which isn't very attractive. And so more and more people are wanting single oysters and using microculture. So there's a lot more to it in a way. The PowerPoint that is on our website is pretty kind of concise about different things that we haven't gone over. But that's the general gist of what's going on. And I really encourage you to come to the Marine Center and get your feet wet and to get used to what we're doing. We still will be dabbling with some scallops. We're done with our oysters. I think we have a good batch. So we'll be now just getting everything into the nursery and getting ready for everybody. Tiana is going to be coming online shortly. The docks are going in. I see Janet's there. Hi, Janet. So anyway, I think we're going to have a great 2022. And
Oh.
hi there. I encourage folks to come out to Ram's Head. I know it's a hike. I'd love to get a shuttle, but we'll figure all that out. There's no hard sell for going. We'll be there all season long, but I think that'll be fun. And we'll plot out anything that people want to do. You know, we're talking about doing a culinary session in June. So maybe we can coordinate that so we can get a bunch of people in their kitchen and we'll all do this kind of cook-off, Zoom cook-off. And then, again, this whole COVID thing just is tenacious as the day is long. I don't know when people are going to be feeling comfortable about. People still come to SPAT. Nobody ever stopped coming there. There's no mask mandates. Everyone's been safe. So you're more than welcome to come there. There's no real restrictions at this point. That seems to be changing all the time. I can't keep up with it. It's a real bore, but I'm vaccinated. I feel okay. I haven't it's funny. I haven't gotten sick in years because, we don't hack on each other anymore. And my wife, who was a school teacher, wears a mask all the time. So she hasn't gotten sick for the first time in, 28 years. So anyway, I look forward to seeing everybody, and it's going to be a good year. And if anyone has any questions, otherwise, I'll let you get to your dinner, and weekend's coming up. If you want, by the way, tomorrow shucking lesson is 100 happy oysters that were donated, and there's no charge. So if you want to eat oysters, that'll probably pay for your ferry, just eating oysters as I'm shucking them. So you're more than welcome. And we'll do that more than once. There's definitely going to be opportunities for shucking lessons. I'm thinking of making a new video. I made a video, but it was really lame. So I'll make a better video on shucking lesson coming up. Good. Excellent. Good. Well, let's stay in touch and have a great evening. And if you need me, I've always got my phone on and I've got my email. So there's plenty of ways of getting in touch. Good.
Thanks, Kim.
Bye.
You're welcome.
I'm going to
Thank
stop
you.
recording.
Thank